بسم الله الرحمن الرحيم. أبدأ معاكم our second lecture dealing with the gram-positive bacteria. Today we're going to deal with the first group of gram-positive bacteria, including the pyogenic cocci. Pyogenic, of course, means pus production, or pus producers, and cocci that their shape is spherical. This group includes Staphylococcus, Streptococcus, and Enterococcus. Today we're going to deal with Staphylococcus organism. This group of organisms have certain properties. They are first extracellular. معناها it means that when they enter our body, they are present outside the cells. They do not exist intracellularly. The other types of bacteria we're going to deal later on, they exist in our body intracellularly. But this group, as we said, they are extracellular and they have high ability to spread in our body tissue, within our body tissue, so they have very high ability to invade our body. So by this we call them, وبالتالي we call them extracellular invasive pathogens. Of course they produce exotoxins. زي أي نوع من أنواع gram-positive bacteria كتير منهم they can produce exotoxins. But the main cause of their invasiveness and growth within tissues is due to their ability to produce a big amount of enzymes rather than the production of toxins. Of course they can cause acute infections that involve the formation of purulent exudates. إفرازات بتبقى اللي فيها الصديد أو البص. ودي usually is accompanied by fever. Let's start to speak بقى إن إحنا نبتدي نتكلم to speak about Staphylococci, the genus Staphylococci. As it is of this group, so of course it is able to produce pus, as we said before. In fact, Staphylococci are responsible for more than 80% of all suppurative infections. والكلمة, this word means برضو infections producing pus. But as you see here, we say here, or here is written, they are almost always. Not always, but almost always. فهنشوف في بعض الانفكشنز بتاعت الستافيلوكوكاي إنها بتعمل برضو non-suppurative infections وبيكون السبب the main reason for this non-suppurative infections هتبقى due to إيه؟ Due to the toxins rather than the enzymes. The morphology and metabolism of Staphylococcus. This member of the genus Staphylococcus appear as a mass of gram-positive cocci. The word Staphylli is a Greek word, كلمة جريجي أو لاتينية يعني بمعنى a cluster of grapes. وده الشكل أو الارrangement بتاع الأورجانيزم لما نشوفه under the microscope بنشوفه as a cluster of grapes زي ما is clear in this picture. They are non-motile. They do not possess flagella, so they are non-motile. They do not form spores, and they are facultative anaerobic. With the word facultative anaerobics means that it's mainly aerobic bacteria, but it can sometimes تستحمل أو withstands an anaerobic condition. So it is mainly aerobic bacteria. وبالتالي it gives a positive, catalase positive, and other features of this organism, الميتابوليزم بتاعها, it ferments sugar to form acid but no gas, and it grows well on any routine media. The most meat infusion laboratory media زي nutrient agar أو nutrient broth, it can grow well on these media. It doesn't need enrichment media, for instance. The classification of Staphylococci. Staphylococci, this is the name of the genus. If you want to know the different species, there are more than 20 different species. But only 3 are mostly associated with the human disease. Their name are Staphylococcus aureus, and as you can recognize here, we always write the genus with a capital letter and the species with a small letter. And we are supposed to always write also the name of the organism in italics. So we have the Staphylococcus aureus, which is the major pathogen, Staphylococcus epidermidis, which is a normal flora of the skin, and Staphylococcus saprophyticus. Saprophyticus comes from saprophytic, so it can be present in any non-living places. The three of them can cause disease to the human being, but the Staphylococcus aureus is the major pathogen. So we will concentrate only on Staphylococcus aureus. A classification of Staphylococcus aureus. There is something called phage typing. By this phage typing, they were divided the Staph aureus to 4 main groups, and they are given the letters or the numbers, the letter numbers, group 1, 2, 3, and 4. How can we do this test? We get a seeded agar plate seeded with the unknown strain of the Staphylococcus aureus, and then we add drops. Here this plate is full of many drops, but if you want to differentiate between our 4 drops, so we're going to make only 4 drops of the different phages. What is a phage? Is the bacteriophage, is the virus that can infect the staph. So we're going to drop the 4 different phage groups on the surface of the agar plate, اللي هو seeded with the test organism, and then it is allowed to be incubated for 24 hours at 37 degrees centigrade. If the phage is specific to the organism, it will cause lysis of the organism. So we will find these clear plaques. This clear plaque, this means that the bacteriophage was able to cause lysis to this strain, so it is specific to it. So we are going to say that this is, for instance, if the phage we dropped here is phage number 1 and it caused the lysis of this strain, so we can consider it to be phage number 1. It is specific to it. The pathogenicity of Staphylococcus aureus. The ability of Staph aureus to produce disease depends on its resistance to phagocytosis and its production of extracellular toxins and enzymes. But what we will deal with these sources of virulence of Staph aureus. Number 1, cell surface antigens. There is something called protein A, a substance موجودة على السيرفيس, على cell wall of Staph aureus. 90% of Staph aureus possess this cell wall protein. Only present in Staph aureus, you have to know this information, is only present in Staph aureus, characteristic for Staph aureus, which is covalently linked to the peptidoglycan. This protein A has a very unusual property, which is binding to the Fc region of the normal immunoglobulin G. We all know, of course, that immunoglobulin, like this little figure, it has the Y-shaped structure. Any antigen is supposed to attack or to bind in this part of the immunoglobulin G, مش في الFC region. But here we can find that the protein A binds in a opposite direction, so it can bind to the Fc region of the normal immunoglobulin G. Not only that, but each one molecule of protein A can bind to 4 of these immunoglobulin G. So they block the immunoglobulin G. And by this, we can read it here, the protein A has the unusual property of binding to the Fc region of the normal immunoglobulin G. It has 4 Fc binding sites on each molecule. It has been suggested that this property imparts an antiphagocytic activity by competing with the leukocytes for the Fc portion of the specific opsonine. Because we all know that the leukocytes are supposed to bind at this direction or at this site. So when this protein A comes and attack or bind to this site, it will block the immunoglobulin G. second virulence of this organism is the presence of a capsular polysaccharide A. But it's actually a microcapsule only visualized by electron microscopy. It is a substituted ribitol phosphate teichoic acid. It is released in the environment and it protects the organism from opsonization by depleting a complement. C. A clumping factor. A clumping factor, this is a protein present at surface برضو of the Staph aureus. الفانكشن بتاعه ان هو can bind to fibrinogen. So this will help the Staph aureus to aggregate. يخلي الستاف aureus تتجمع to form aggregates. طبعا aggregated Staph aureus are difficult to be phagocytosed. D. Binding sites for a number of serum proteins. This character is not present, or this property is not present in every strain. But if a strain contains a binding site for fibronectin, for example, which is a kind of a serum protein, this fibronectin, and it is found in places of the wound. It's usually found in places of the wound. So wounds contain much of this fibronectin. So this can explain why there is persistence of Staph aureus in wounds. Because if it has this receptor to bind to the fibronectin, we will find that this organism will persist in the place where there is a wound, and of course it will lead to wound infection. فده ممكن يشرح لنا, explains why Staph aureus cause wound infection. There are other serum proteins, for example, collagen. Collagen is present in bones. So if the Staph aureus has a certain receptor to bind to collagen, it will cause infection of the bones and it will cause osteomyelitis. ننقل بقى للحاجة التانية, extracellular enzymes. طبعا الستاف aureus can produce مجموعة كبيرة من extracellular enzymes. أول حاجة coagulase, which is very specific for Staph aureus. What does this coagulase do? الميكانيزم بتاعه إن هو converts الفايبرينوجين to fibrin. يعني معناها it makes blood clot. But it needs the help of the clumping factor. احنا قلنا the clumping factor causes the aggregation of the Staph aureus together, and then the coagulase converts this fibrogen to fibrin, اللي هي blood clot surrounding the organism. Of course, this will inhibit phagocytosis. Afterwards, الهيموليزنز. هيموليزنز من النيم بتاعها ان هي بتعمل lysis لل RBCs. There are 4 such toxins produced by Staph aureus, but they are different in their structure, in their specificity, and even in the mechanism of action. So we call them alpha, beta, gamma, and delta. But all of them بيتجمعوا مع بعض في إنهم إيه؟ Have the same character إن هم can destroy red blood cells. For example, alpha hemolysin causes lysis to the rabbit RBCs and the human platelets. Okay? C. Panton Valentine leukocidins. Panton, the word Panton and Valentine are the names of the scientists who discovered this enzyme. So that فاتسمى على اسمهم Panton Valentine leukocidins. Leukocidins, they cause the destruction of the white blood corpuscles أو damaging the white blood corpuscles. They are composed of 2 components, S and F. They act synergistically to damage the white blood corpuscles. Hyaluronidases. واحنا جبنا سيرتها in the introduction. وكلهم جبنا سيرتهم in the introduction. Hyaluronidases is called also spreading factor. This enzyme hydrolyzes hyaluronic acid, which is a constituent of the ground substance of the connective tissue. So helps the bacteria to spread in the body. So that's why they call it spreading factor. Lipases. Lipases, from its name, digests fat. And they are believed to be an important very important in the pathogenesis of boils. Boils actually is formed of the destructive substances of the body, like the fat, like the protein, the DNA, the RNA, all of these substances when they are destroyed and يحصل لهم lysis بيتحولوا إلى البس اللي بيكون البويلز, which makes the boils. Staphylokinases or fibrinolysins. Here we call this, we say this is encoded by bacteriophage. And there must be a bacteriophage infecting the organism, our bacteria, so that they can produce this enzyme. It converts plasminogen to plasmin. And this plasmin is capable of lysing fibrin, so helps in the spreading of the organism. So we can see here that it do the function, the opposite function of the coagulase enzyme. The coagulase causes coagulation of the organism and covering it with the fibrin, and then when the organism needs nutrients, for instance, and it wants to leave this يعني escape بقى من الحبسة بتاعت الفibrin, so it produces this fibrinolysin which will dissolve the fibrin, and so it can get free again and move and spread in the body. Several proteases and nucleases, these cleave the proteins. The proteases, of course, will cleave the proteins, and the nucleases will cause the destruction of the RNA and DNA respectively. Beta-lactamases, these are also enzymes produced by this bacteria, which have an important role in the antibiotic resistance. And of course, you have taken these enzymes in the antibiotic course last year. Finally, we're going to speak about the exotoxins produced by this organism. مش كل species can produce the exotoxins. فهنشوف مثلا the first type of exotoxin is staphylococcal enterotoxins, وبيترمز لهم SES. These staphylococcal enterotoxins برضو are carried by bacteriophages اللي هي احنا سميناها ايه lysogenic. We explained this in the introduction لو تفتكروا. قلنا في حاجة اسمها lysogenic, يعني الجين to produce this enzyme comes from a bacteriophage. يعني it comes from the virus that attacks the organism. And it's introduced into the staphylococci via a phage conversion. اللي هي through the lysogenic life cycle. It produced by only the phage group 3 Staph aureus. مش احنا قسمنا we classify the Staph aureus to phage types. So phage type group 3 هو can produce this toxin. This toxin has a very unusual property. We all know that exotoxins are protein in nature, and they are supposed to get denaturated by boiling. But we find here that this toxin resists proteolytic enzymes and also resists a high temperature and can withstand a high temperature up to 100 degrees for 30 minutes. There are 7 staphylococcal enterotoxins. Staphylococcus enterotoxin A is the most often encountered in food poisoning, while Staphylococcus enterotoxin B plays a role in toxic shock syndrome. Of course, we're going to deal with this type of syndrome later in this lecture. And it is believed to be a super antigen. طبعا the word super antigen, we don't still know what is the meaning of a super antigen. أنا أحب إن أنتوا تدوروا عليها وتبحثوا فيها وتشوفوا معنى super antigen لغاية ما أنا أشرحها لكم إن شاء الله in the next lecture. Exfoliative toxin. Exfoliative toxin, these are produced by 5% only of the Staph aureus isolates, mostly in phage group 2, and they are sometimes called epidermolytic toxin due to their action. لأن الأكشن بتاعهم إنهم بيعملوا عملية تقشير أو peeling of the skin. So that's why they are given this nomenclature, exfoliative أو epidermolytic toxin. And they cause the disease or the syndrome known as staphylococcal scalded skin syndrome, and they also have an effect on the disease called impetigo. C. Toxic shock syndrome, toxin 1. It's also believed to be a super antigen like the Staphylococcus enterotoxin B. And they are responsible for the toxic shock syndrome. And it is believed that this organism is produced when the gene of the production of this toxin is activated by change in the pH, in the acidic side, to the acidic side. Yeah. Now we want to deal with the infections caused by Staphylococcus aureus. We say that this Staphylococcus aureus is a normal resident of the skin, nares and bowel of 30 to 50% of the general population. يعني in this organism we can consider it as a normal flora persisting in our body, on our skin and in the nose. Nares, it means the nose, and in the GIT اللي هو large intestine, البول, in 30 to 50% of our population. So of course they live بما أن هم, as they are a normal flora, they must live without harmful effect. So they live in these areas without harmful effect. ولكن under certain conditions they begin to penetrate the unbroken skin, and this is important. They can have the ability to penetrate unbroken skin, or through the hair follicles, or they can pass through the ducts of sweat glands, and then causing extensive disease. طب what makes them يتغيروا from being harmless to being harmful? This infection probably never occur unless يا إما a highly virulent organism is encountered or the body resistance is lowered. So we can call them, in this case, that they are opportunistic pathogens. Staphylococci also are believed to be one of the top 4 causes of nosocomial infections, اللي هي hospital-caused infections, along with Escherichia coli, which is a gram-negative bacteria, of course, Enterococcus faecalis is another gram-positive bacteria, and Pseudomonas aeruginosa is a gram-negative bacteria. So we have here 2 gram-positive and 2 gram-negative bacteria are considered the top 4 causes of nosocomial infections. This is due to what? يعني ليه هم بيسببوا problems in hospitals? أولاً, first, Staphylococci, especially Staphylococci, is resistant to dryness. We call it it's hardy. It can withstand very unfavorable conditions like dryness. Although it is not a spore, but it has the ability to withstand dryness. So easily and rapidly spread to patients from the hands of the clinical staff and from vomitives such as clothes, sheets اللي هي الملايات بتاعت السراير, and any equipment. تاني نقطة, Staphylococcus aureus strains are typically resistant to many drugs and can avoid being killed even by most disinfectants, especially the MRSA strains. تاني انتوا أخدتوا فكرة قبل كده عن كلمة MRSA. MRSA is meaning methicillin resistant Staphylococcus aureus strains. They started by being resistant to methicillin antibiotic, and then they became multiple resistant to many types of antibiotics. Number 3, of course, in the hospital environment, we're going to encounter people who are, of course, not healthy people. So this can make it to pass to individuals who are highly susceptible to staphylococcal infections, including newly born babies اللي ممكن يكونوا موجودين في nurseries في المستشفيات, immunosuppressed patients, burn victims, الناس اللي عندها حروق, patients with indwelling devices, يعني ايه indwelling devices؟ يعني اللي هي الاعضاء الديفايسز اللي بتبقى داخله مش من الجسم. الاندويلنج ديفايسز زي ايه؟ زي الكاثيترز، القسطرات، او البروستاتيك ديفايسز اللي هي زي مثلا البيس ميكر اوف ذا هارت، برضو الهارت كاثيترز، القسطره بتاعت القلب. كل دي حاجات ممكن انها تخش في الجسم وتسبب infection by Staphylococcus aureus. Suppurative infections. Suppurative infection means that these infections will produce pus. أول نوع اللي هي skin infections. في كذا نوع من skin infections actually. Staphylococci are the main cause of suppurative skin and tissue infections. Staphylococci commonly enter the skin via an insect bite or hair follicle, as we said before, and they will form a pus-filled lesion. We call it pustule. This pustule, if it's infecting the hair follicle only, we call it folliculitis. يبقى folliculitis occur when the lesion is restricted to the hair follicle. ولكن لو حصل more spreading للأورجانيزم أو الحالة بقت more severe، ممكن يحصل وممكن ما يحصلش. If it is more severe, we call the lesion furuncle. الفرنكل when the pustule spreads into the subcutaneous tissue. هنشوف دلوقتي we will see a figure to explain the different stages of this skin infection. Then these furuncles may spread beneath the skin and begin to form interconnecting channels to form a complex lesion known as carbuncle, which is often seen in the neck and the upper back. عادة بتظهر في الرقبة وفي الظهر. It may lead to cellulitis, a more severe form of infection, and then bacteremia. The organism can pass and enter to the blood, especially in the immunosuppressed patients. هنا this figure بتوري the different stages of the follicular infection, skin infection. Here this is superficial folliculitis. The pustule is in a single follicle of a hair follicle. هنا الهير follicle اهي، وهنا باين السيلز دي اللي هي الباس اللي موجودة اتكونت in the hair follicle. لو it was more deep، الfolliculitis is more deep اللي احنا we call it بقى ممكن في الستيج الأتقل كمان من كده بنسميها furuncle. هنا زي ما انتم شايفين in this part of the figure this is a furuncle. بيبقى الانفكشن امتدت in the subcutaneous tissue. هنا كانت only superficial فوق in the edge of the يعني على السيرفيس. هنا بدأت تنزل تحت. هنا بدأت more severe infection. الكربونكل بقى بيبقى عندنا more than one head, more than one hair follicle is involved, and we can see that the infection is more spread and it is ممكن تتأدي كمان إلى fever. تبقى systemic infection ويبقى فيه fever، المريض يبتدي يحس بالفيفر. طبعا زي ما قلت مش لازم كل المراحل دي تحصل، ممكن الانفكشن stops at this stage but in severe stages it can توصل للستيج بتاع الكربونكل. هنا شكل تاني another figure أو to show the folliculitis, the simple form. Here is a carbuncle with more than one head. An abscess is seen on the forehead of a 60-year-old man. And here is cellulitis, more severe طبعا واضح قوي. It's very clear and obvious from the picture that the case is very, very complicated. ننقل بعد كده we go to impetigo. Staphylococcus aureus is also a major cause of impetigo. Impetigo in Arabic means الحصف. It occurs most often in young children living in warm climates. يبقى it بتحصل أكتر في الجو الحر. الانفكشن is promoted by poor hygiene and crowded living conditions. يعني ممكن تحصل في فصل مثلا مدرسة in a school classroom where it is overcrowded and there is no good hygiene. فيبتدي تنتشر this skin infection between the pupils in the room, between التلاميذ. بتبتدي منين؟ It appears as thin-walled vesicles زي ما هي واضحة. It's clear on the picture here. Thin wall vesicles on the skin, most often around the nose and then spread over the face. وممكن الأورجانيزم يكون برضو as a normal flora in the nose. والطفل الchild ممكن is suffering from running nose, so the running nose will infect his face around the nose and then spread all over the face. These fine walled vesicles then rupture and then forms a crust, قشرة، بيكون قشرة على lesion, then later crust over. These are the skin infections caused by Staphylococcus aureus. We can have a treatment for the skin infection, for general skin infection. This is not only for impetigo, this is a general treatment for skin infection. First of all, we have to drain the pus from the lesion because any antibiotic will not be able to penetrate through the abscess. Then we will apply a local treatment, a topical treatment like mupirocin antibiotic. It's a drug of choice for skin staph infections. Or fusidic acid is also successful for treatment of staphylococcal skin infections. If there is systemic effect زي ما قلنا in the carbuncles أو في cellulitis، ممكن في الحالات دي بيبقى فيه ارتفاع في درجة الحرارة، بيبقى فيه fever, and we need to take a systemic treatment. So we can use dicloxacillin or erythromycin. For example, and these are examples I'm giving to you. It's not يعني مش شرط لازم this antibiotic to be used. طبعا it's better to use to apply antibiotic sensitivity test and choose the most appropriate antibiotic for the case, for each case. Now we're going to speak about pneumonia. Pneumonia is a secondary infection in our case. When it comes from Staphylococcus aureus, it is a secondary infection. 75% of the patients are under 1 year of age. Mean are under risk to get this secondary infection of pneumonia. يعني it starts مثلا for example with a viral infection, respiratory infection, and then الستاف هتتاخد بتبقى opportunistic في الحالة دي، تنتهز الفرصة and it gets this kind of pneumonia. So عشان كده we call it a secondary infection. يعني it usually doesn't come to a person without any infection. يعني هو يكون عنده أصلا infection في respiratory system، مثلا is suffering from measles, suffering from influenza virus, and then it is complicated with the pneumonia caused by this organism. مين اللي هم under risk؟ those with impaired immunity, or زي ما قلت pulmonary infections, such as those with cystic fibrosis, measles, influenza, and those receiving chemotherapy. This kind of pneumonia is acute and frequently fatal. We can see in this figure the normal alveoli, and here is the alveoli suffering from pneumonia. You can find the yellow color here is due to the pus and the suppurative exudates اللي بتبقى موجودة in the lungs. Number 3, osteomyelitis اللي هي infection of the bones. واحنا جبنا سيرتها قبل كده، قلنا الاورجانيزم اللي عنده a receptor to collagen هو اللي في الغالب can cause this kind of infection. Osteomyelitis is infection of bones occurring هنا most frequently in boys under 12 years of age. اشمعنى boys under 12 years or age are more susceptible to this kind of infection? They say because they are the age where they always play, for instance, football, or in any playgrounds, and they are liable to get injured, to get fractures in their bone, or to get any kind of injury. So the organism may lodge on an injured or broken bone or burned area, but it must be third-degree burn, because third-degree burn destroys the full thickness of the skin, so can reach easily to the bones. Or it occurs in patients who had orthopedic surgery, يعني surgery in their bones. And these are pictures of legs with هنا there is osteomyelitis in the left leg. I don't know if you can read the writing here, it's a bit small. In the tibia, يعني هنا tibia is infected with the Staph aureus in these legs. Endocarditis. The organism enters and reaches to the heart through bacteremia. يبقى البكتيريا is present in the blood, and from the blood it reaches to the heart. If this occurs, it occurs to a normal heart, so it is a primary infection, and it can lead to endocarditis, which is acute and rapidly progressive, that can destroy the natural valves within days. And it is very severe. It has a high fatality rate from 40 to 80% even with treatment. So endocarditis caused by Staph aureus is very severe and it is fatal. Number 5, enteritis. You can recognize that this organism, when enters to any place in the body, it can cause infection. A suppurative infection, of course. Enteritis, severe infection of the small or large intestine. This can develop after GIT surgery or after intensive antibiotic therapy. If this antibiotic destroys the normal intestinal flora, as we said before, and a normal flora protects us from any outsiders, other invaders. So if the antibiotic destroys the normal flora, this organism persists in the GIT and begins to cause supra growth and then supra infection, especially, of course, if the drug-resistant Staph aureus is encountered, like MRSA strain. So it causes enteritis. Wound infections. It is less common normally كده في الحياة الطبيعية in normal life, but it's more منتشرة, more frequent in nosocomial staphylococcal infections. In hospitals, the wide use of antibiotics, many of which are bacteriostatic, has resulted in the emergence of antibiotic-resistant strains اللي هي اتكلمنا عنها قبل كده we dealt with before اللي هي الميرسا strains. طبعا these which may spread to other patients in the wards and to the hospital staff. يبقى it is a big problem اللي هي wound infection in the hospitals. And you can see this figure. This is a wound وبدأ فيه pus يتكون إن حصل إيه؟ حصل infection with staph وبدأ يحصل pus formation and abscess in the wound. In addition, we can say that Staph aureus infections may include septicemia. The organism passes in the blood and goes and spread to any part of the body. And this we call it metastatic abscesses, forming metastatic abscesses. Example, it can enter the kidney, cause pyelonephritis, to the brain, cause brain abscesses or meningitis. Now we're going to deal with the non-suppurative infections due to Staph aureus, or we can call them the toxigenic diseases. يعني هنا the main cause of these diseases are due to the toxin production. Number 1, staphylococcal scalded skin syndrome, and we give it the abbreviation for S. This is an acute exfoliative dermatitis characterized by wrinkling and peeling of the epidermis. The body looks like as if it is scaled, or the skin is scaled with boiling water. As we see in this picture, the skin of the baby حصل له peeling as if you have poured boiling water on his skin. But for good sake, only the granular layer is involved. Thus, staphylococcal scaled skin syndrome lesions heal without scarring, and it doesn't leave any scar on the skin. And this healing takes from 7 to 10 days. This syndrome, as you can see in the pictures, is usually seen in young children under the age of 2 years. This case is caused by phage group 2, of course, which produces the exfoliative cytotoxin. Toxic shock syndrome. Toxic shock syndrome, and also we give its abbreviation TSS, was first discovered in the year 1978. At that time, 96% of the reported cases were women who were using super absorbent tampons during their menstrual cycle. These tampons, of course, are inserted inside the vagina to prevent the blood to come out from the bleeding to come out of the body. So it's a very good media for the growth or even the overgrowth of the bacteria, and then it begins to produce this toxin. The pH بيبقى acidic, so this toxin, as we said before, is بيتنشط الجين ده is activated by the low pH, so it begins to produce the toxin. However, it is not only a disease of women. لازم نحط في اعتبارنا انها مش بس women which get this infection. It may also occur in individuals who have any focus of staphylococcal infection. Any other focus of infection ممكن برضو يبدأ ويسبب this kind of disease اللي هو toxic shock syndrome. Toxic shock syndrome is caused by Staphylococcus aureus strains that produce التوكسين اللي هو احنا قلنا عليه in the beginning اللي هو toxic shock syndrome toxin 1 or Staphylococcus enterotoxin B. The symptoms of this disease are fever, pruritus. Pruritus يعني الجلد بيبقى بيهرش، بيبقى فيه itchy sensation in the skin. There is rash, and طبعا you can see the rash very obvious in these two pictures. بيحصل desquamation of the palms of the hand، الكف بتاع الإيد، and the soles اللي هي قعر الرجل، وهنا باينة الصورة دي in the legs هي the soles and بيحصل فيها الإيه؟ desquamation. There is also nail and hair loss. المريض بيبتدي الnails بتاعته becomes very weakened and he can lose it, and the hair the same goes for the hair. Extreme fatigue, تعب شديد, and systemic signs of toxicity including hypotension and shock. الحتة دي is very important, this part اللي هي signs of systemic toxicity. If you can remember that I said that these two toxins are super antigens. So super antigens من ضمن الكاركترز بتاعتها اللي احنا هنعرفها in more details the next lecture inshallah. They cause hypotension and shock, that this is a systemic sign of زي pyrogenicity. And if you can remember again in our introduction in the table when we compared exotoxins and endotoxins, we said that exotoxins may produce pyrogenicity, and here this هنا بقى بتبان حكاية الهايبيروجينيستي إنها بتسبب الهايبوتنشن and shock، والرينج بتاعها أو الريت بتاع المورتاليتي ريت هنا بتبقى from 5 to 10%. And as I said, super antigens, I wish that you can look for it, research for the meaning of a super antigen till I explain it to you next lesson inshallah. We can see here more pictures about toxic shock syndrome. Here are the palms of the hand peeling of the hands. We see here the nails are very brittle or very weak, and they can يحصل loss of these nails. And the rash, this picture is not very clear. A rash is more clear in the previous picture. Number 3, food poisoning. Staphylococci cause one of the most common forms of food intoxication. طبعا كلمة the word intoxication means that the food contains the toxin already. يعني الorganism already elaborated the toxin in the food. So the person eats the food, ingests the food containing already the toxin. So we call it intoxication. The signs here is very quickly appears within 1 to 6 hours. Even within sometimes half an hour, the person begins to feel the symptoms, how to get the symptoms. After the patient has eaten a protein-rich food. Example of this protein-rich food, cream-filled pastry زي التورت مثلا اللي بيبقى فيها كريم. الchicken salad, for instance الchicken salad اللي هي بتبقى فيها مايونيز وبتبقى cold, served cold, and cottage cheese اللي هو زي الجبنة القريش. يبقى دي examples of food which can be intoxicated by this toxin. So they are already infected with the enterotoxin producing Staphylococcus aureus. The enterotoxins are heat stable, and this is something very characteristic لأن احنا عارفين إن زي ما قلت قبل كده إن exotoxin must be heat labile, but here this is heat stable, and the food كمان doesn't appear spoiled. ما يبانش على الفود إن هو فيه أي حاجة غلط أو إن هو بايظ أو كده، فالشخص هياكل الفود وهو مش حاسس بأي حاجة، وبعدين يبتدي يظهر الساينز أو السيمتومز very quickly within 1 to 6 hours. إيه هي السيمتومز؟ severe vomiting and diarrhea. الvomiting بيبقى أكتر حاجة characteristic for staphylococcal food poisoning is the severe vomiting because it affects the vomiting center in the brain, in the CNS. And diarrhea lasting from 4 to 48 hours. It is not a very dangerous food poisoning actually, but it's only life-threatening for infants and elderly. Okay, but for an adult, it is not that severe kind of food poisoning. Here we want to know the general treatment for Staphylococcus aureus infections. We can choose methicillin or oxacillin, which are known to be penicillinase-resistant penicillins, but they are only active, of course, for methicillin-sensitive Staphylococcus aureus. If the case is MRSA strain, we have to use a more powerful and potent antibiotic like vancomycin or clindamycin. However, it is more advisable to undergo antibiotic sensitivity test before choosing the proper antibiotic. In case of toxic shock syndrome, we have to treat the severe drop of blood pressure, so we have to have supportive measures for the shock. And in case of food poisoning, the antibiotic is of no need, as we said, it is due to a toxin. The only treatment is that the patient should only be adequately hydrated by taking fluids to prevent dehydration of his body. دلوقتي ننقل بقى to the laboratory diagnosis. We isolate the organism from the infected lesion. If it is a skin infection, هناخد عينة, a sample from the skin lesion or from the blood samples in other cases. And we're going to cultivate it as we اتفقنا من أول إنها can grow on or the normal media اللي هي nutrient agar media. So we're going to cultivate it on the nutrient agar plate. We will observe the characteristic golden color of the Staph aureus. يعني أنا يمكن نسيت أقول لكم that the word aureus means gold. So it's the golden color of the organism. The colonies of the organism, when we grow it on the nutrient agar, it appears yellow in color. Somehow we can consider it as golden in color. So we can observe the golden color and the shape, of course, of the colonies. اللي هي pinpoint head. I think you will remember this from your lab sections. It's by gram staining. We can, of course, detect it as a gram-positive. We can see it under the microscope as a gram-positive spherical clusters. اللي هي grape-shaped clusters. حاجة بقى selective for Staphylococcus aureus, and we cultivated on mannitol salt agar. Mannitol salt agar has a high concentration of sodium salt, so that it can be selective for staphylococcal species. But when we grow on it Staph aureus, the color of the mannitol agar, which is a red color, changes to the yellow color. Of course you have taken this in برضو في اللاب، ففيش داعي أضيع وقت في شرح المانيتول salt agar. Then coagulase test. There are two types of this test: tube test and slide test. في التube test, of course, it is applied in a test tube. من هنا جت nomenclature tube test. It detects the free coagulase, and this test needs a serum prothrombin to act. عشان الرياكشن تحصل بنحتاج لسيروم prothrombin. فwe get citrated rabbit plasma ونعمل لها inoculation with the organism, incubation at 37 degrees centigrade. زي ما قلنا the plasma will contain the fibrinogen اللي هي هيتحول مفروض to fibrin. So if the staphylococci produce the free coagulase, which is secreted outside the cell, عشان كده سميناها ال free coagulase because it is secreted outside the cell, هيحصل ايه للرابت plasma? It will change from the liquid form to a jelly form, and this takes within 1 to 4 hours. And we can see here the picture. This is the positive jelly formation in the test tube, and this is the negative test tube, the control negative. The other test is the slide test, and here we detect the bound coagulase. The organism is mixed on a slide with a loop full of human plasma. If it is coagulase positive, بيحصل clumping of the cells, which occurs in few seconds, and this is due to the clumping factor. And we can see here the slide. Of course, هنا حصل a clumping, and here this is a negative result. It should be pointed out that although the presence of coagulase enzyme provides a good evidence for potential pathogenicity of the staphylococcal isolate, but its absence doesn't mean or doesn't indicate there is lack of pathogenicity. بمعنى ايه؟ ان ممكن الانفectious agent هنا يكون staphylococcus other than staphylococcus aureus, and it is still pathogenic, and it is still the causative organism, but it gave coagulase negative because it is not Staphylococcus aureus. Okay? Here is another test, and it's usually used to detect MRSA strains. We call it the dry spot Staphytect plus test. The Staph aureus can be confirmed by testing colonies for agglutination with the blue latex particles coated with porcine fibrinogen and rabbit immunoglobulin G and specific polyclonal antibodies. كل دول بيبقوا together في reagent جاي جاهز، وبيتحط على الكارد. هنا بيبقى فيه ورقة كارد كده بيتحط عليها الرياجنت والمايكرو اورجانيزم، فبيحصل الاجلوتينيشن appears on the card. فهنا هنا ده عبارة عن control negative وده التست organism. فهنا it's clear هنا that we have 2 positive and 1 negative agglutination. طبعا الفايبرينجين والاميونوجلوبولين جي والبوليكلونال انتي بوديز المفروض بيكشفوا ايه او بيعملوا agglutination with what in the bacterial cell. الفايبرينجين will bind with the clumping factor in the organism. Immunoglobulin G will bind with the protein A present in Staphylococcus aureus, and the specific polyclonal antibodies will bind, of course, with the capsule, the polysaccharide capsule of the Staph aureus. So when this reaction occurs, the clumping appears, or the agglutination appears on the card. Finally, we want to know how to differentiate. This is a table to just summarize how to differentiate between Staphylococcus aureus and other species of staph. So for hemolysis, we find that Staph aureus causes beta hemolysis. Other staph doesn't cause beta hemolysis. What is the meaning of beta hemolysis? Beta hemolysis means complete hemolysis of the RBCs. Coagulase test, it is positive with Staph aureus and negative with the other Staph species, because we said before that this enzyme is only produced by Staph aureus. Protein A is also a surface protein only present in Staph aureus, so it is present in Staph aureus, negative in other Staph species. And here the mannitol test بيحصل color change in Staph aureus إنه بيتكون it changes from red to yellow. In other Staph species there is no such change in color. The Staph can grow on the media, but there is no change in color. It stays red in color. Finally, بقى prevention and control of Staph aureus. There is no vaccine available until now. طبعا في trials كتيرة قوي الفاكسينز، بس لغاية دلوقتي ما فيش حاجة أخذت الشهادة أو يعني it is approved to be taken as a vaccine. Control is directed primarily at those individuals who are most susceptible to staphylococcal infections, especially in hospitals. Thus, the newborn nurseries of hospitals and surgical operating rooms are particularly dangerous areas where rigid aseptic procedures must be followed. And this is the only way to prevent this organism until now, especially it is a normal flora. Okay, thank you, and نتقابل إن شاء الله the next lecture بإذن الله.